All right. Hello, everyone. Thank you so much for being here with us today, live or virtually afterwards. We are so excited to get to take a look at what these amazing biodesign teams have done over the last few weeks and get to hear all about their projects. Right. So this is I believe our fourth Idea Accelerator cohort of the year already. Wow. It's flown by, and it's been so great to see so many great ideas throughout the year. And this is part of the broader BioBuilder program, as you all know, which our real goal is to inspire students to be excited about biology, biodesign, bioengineering, and get to think about what they could do to change the world with biotechnology if they had all the power of biotechnology at their fingertips. Within that, we try to provide a comprehensive synthetic biology curriculum looking at designing, building, and testing different systems where students can integrate these biology and engineering practices through hands-on lessons, club activities, and internship experiences. And teachers, we hope to help you learn new methods to teach and engage students and inspire the young scientists in their classroom. And we have a variety of programs that help us do this. One of our primary ones is the Idea Accelerator, which is the reason you're all here today, which is a 3-week program where students learn the core principles of biodesign and synthetic biology and then get to use them to create some cool biotechnology with a design project. We also offer the BioBuilder Club, which is a year-long program where students often will delve into the build and maybe even test phases of this biodesign project, as well as some advanced seminars and other kits and programs. And we will start off with the first team from the Washburn School today. I'm very excited to hear what the Washburn ProCo team did over the course of their project.
Renewable fuel. We are the ProCo team from Washburn High School. Our group consists of Katie Ritter, Bailey Brown, Zavoy West, Kason Isham, and Taylor Middleton. The topic area we decided to work on was fuel. Initially, we had other ideas related to medicine. We decided to focus on diesel fuel because it would be beneficial to the world and it was also easier to work on.
Reasons it would be helpful. The system we designed should be a second option for the limited supply of oil. The input could be E. coli bacteria and the outputs could be cheaper and more oil for the fuels. The next step to figure out is to make the E. coli sugar turn into oils for renewable fuels. Renewable fuel would be helpful because it would make jet fuel much cheaper. Also, with renewable fuel, it would be easier to get fuel. This would eliminate the problem with having to trade with other countries. The bacteria that will be used is called E. coli. There has been genetically modified versions of E. coli bacteria that can convert sugar into the oil that is needed for fuels. It is completely identical of the oil that is already used for the fuels. It converts the sugar known as glucose into the oil that is needed to be used in the fuels.
All right, I think that that wraps up our first presentation. Great job, Washburn ProCo. Awesome project. So excited to hear what you've talked about here today, and absolutely renewable fuels is a huge area of investment and research, and being able to create fuels that that can be used in existing systems and work with existing infrastructure is absolutely a huge area of interest.
Yeah. Michael, can I add just one thing? Because I completely agree. There's that company Amyris out in California that found that. And I think they found it the hard way. They were trying to just come up with fuels that were completely different and then realized that it would actually be a better company if they found a different way to produce fuels that could go into existing systems. And one thing, they actually said it in this presentation that the oil that would come from this bacterial system would be the same as the oil that exists from, digging up the petroleum that we've got in the ground. So, I think that's a really important point that a molecule is a molecule, however it was made, whether it was by chemical engineering or biological engineering. So I think they're really, like keying in on a super exciting idea. If they were going to pursue this for longer, I might ask whether E. coli is the right chassis, because fuels can kill E. coli pretty quickly, the product, but there could be a way to re-engineer that, or to find a different chassis that's better tolerant for some of these fuels. But I think it's a great idea. I love that.
Absolutely agreed. All right, and anyone else in the audience as well, if you have questions that you'd want to pose to the Washburn teams, even though they're not here at the moment, or any other comments you'd like to make, we'd love to hear from you. All right. And with that, we'll move on to the Washburn Fantastic Five. Excited to hear what they've been working on.
Hello, we're the Fantastic Five group from Washburn School. This is Harley Cornell, Kinley Brown. I'm Natalie Harville,
Riley Kaufman, Madison Woodham.
and we've decided to do dairy allergens. So our input is modifying dairy allergens, and our output is to make allergy shots last longer so they could be more infrequent. Topic area we've decided to work on is biomedicine. Initially, we had other ideas related to medicine, but we decided to focus on allergies, specifically in modifying dairy allergens. We want to help people with the allergies by modifying their allergy shots in order to make them last longer. System design. The system we designed should allow people to take less shots for dairy allergies. The input could be a device to detect when more doses of medicine is needed, and the output could be that the device actually adds more medicine when needed. The next step to design this system is to figure out what coding will be needed to produce a device to detect the dosage needed. We want to modify the shots of dairy allergens. Our dairy allergen is IgE. Normally, these shots for the allergen is taken 1 to 2 times a month. We want to get the shots put in the allergen IgE and modify the shots, putting a healthy bacteria or probiotics in its doses. In this case, the healthy bacteria is lactobacillus, and it lasts between 1 to 3 weeks. You can take 1 to 2 billion of them daily. They are healthy because they help break down food, absorb nutrients, and fight off bad organisms that may cause diseases.
All right, fantastic job, Fantastic Five. What great work. Oh, so great to hear about such an innovative project too. I really like the idea of being able to take medicines that already exist and used and be able to make them easier to use for the patient, potentially fewer side effects, lower doses, and just generally less hassle for the patient. One thing that I didn't think of earlier when we were discussing this a little bit, but it has a lot of similarities to things like the artificial pancreas that's used to treat diabetes sometimes, where you have the system that can sense the amount of insulin in the blood and then release sense the amount of glucose in the blood and release the correct amount of insulin. And I think a system similar to that, potentially if you could sense lactose in the gut and then release lactobacillus or release the enzymes that you need to degrade it, that would be a really cool new development in the field. So really, really great work.
Yeah. Can I just chime in? 100%. I thought that was so interesting. The engineering of a microbiome, right, is so relevant right now. And lactobacillus are things that we're generally pretty comfortable taking into our body. Anyway, I have yogurt every morning for breakfast. So if we could engineer those in a way to have this diagnostic and treatment that is part of this. And my son went through allergy shots his whole life. They are terrible. This would be a really considerable improvement. And then the last thing I'll say, I loved it when you talked about coding, right? Like that what you need to do is recode this, right? It is a coding language, and we are figuring out how to program the cells to do the things. So awesome project.
All right, great work. All right, and up next is the Washburn High Giraffe. What a fun name.
We are the Giraffes from Washburn School.
The topic area we decided to work on was environment. Intentionally, we had other ideas related to medicine. We decided to focus on water pollution because it is a problem in the world and our team would like to be part of solving.
System design, to add cells to polluted water to see if there is coliform bacteria, then if detected, the water will turn purple.
The input could be add cells to detect if there's coliform bacteria in the polluted water. The outputs could be if the water will turn purple when it is detected in the polluted water. The next step to this design system is to figure out how to get the bacteria out safely without harming the water even more. What we liked best about this work was learning about the new bacteria in the water and bacteria that are harmful that's in the water.
All right, great work, Team High Giraffe. Very cool. Biosensors are so important, and water quality is really becoming so important, and being able to know that the water you're drinking is safe and clean. And a lot of times there are a lot of bacterial contaminants. So having a system that would be maybe an at-home diagnostic or used in water processing facilities for something like this would be a really great development. Having sort of a nice, colorful output would be fantastic. I really like the clear input and output here of like, we want, the input is the bacteria, the output is this color that we can see that will tell us that the input is there.
That was, I was going to say so many of those things too, Michael. I love the input output. I hadn't really thought about an industrial application for this. I was just thinking of an at-home diagnostic, maybe because, you know, I like to spend time on a lake in the summer, and there definitely are coliform, you know, contaminants in it from the ducks and everything else that land on that lake. And I guess I was thinking not of putting the cells into the lake, but actually taking some of the lake water out and using this engineered system as a test, like a little testing kit, almost like they do in the pool. Like if you're swimming in a swimming pool, I think that could be a really straightforward form factor for this experiment or this technology. And yeah, input output is all defined. So just building it.
Yeah, absolutely. And I love the idea of sort of taking this system too and using something existing like a pool pH monitor that people already know how to use to really make it something that would be an adaptable technology
for sure.
All right, up next is the Spice Girls from Washburn.
Here's the Spice Girls team from Washburn High School.
The topic idea we decided to work on was food. Initially, we had other ideas related to medicine.
We decided to focus on food because we wanted people to be able to live a healthy life while enjoying the flavor of junk food. So picky eaters like kids not liking vegetables will eat them because they taste like chicken nuggets.
The system we designed should change flavors and textures of food that taste like something else.
We put biomediating tools and cells. Output: generate better flavors and foods by enzymatic reactions.
Input: enzymes. Output: direct access to a flavor compound.
Input: aromatic scents. Output: trick your brain into thinking you're eating something you're not.
Some of our ideas were using aromatic scents by neutralizing taste buds or flavor in food, then using scents of your favorite food to trickle your brain.
All right, great work, Team Spice Girls. That's awesome. What a clever idea. And
Yeah.
I really like their observation of both the sort of big and small outputs of like, we want the output of the food will taste a certain way, but also like we want the output of people will enjoy the food, and sort of the input of enzymes and the input of the bad flavor being gone. So looking at these different things that they want to come in and out of this sort of complex system that they're developing. And I have even friends my age who I think would enjoy if you could make vegetables taste like chicken nuggets. What a great technology that would be.
Yes, my stomach started rumbling right then. That's for sure. That would be great. I also will compliment this team. Love that the Spice Girls is made up of both genders. I think you guys are very smart and very clever to do that. But I also, like Michael, really feel like you've zoomed out of this system idea to be able to consider many different aspects of it. And I really compliment the team, because a lot of times teams zoom in so fast on exactly what they want to do, and they don't always consider all those different aspects, you know, of the scent and the neutralizing of the existing taste buds and things like that. So I think for an early stage idea for a project, this is great and could go in a lot of really interesting directions.
All right, such great work. I think everyone's on board with this one. Yes, Kelly. Awesome project. All awesome projects so far, really great. And next is the 5 directions.
My name is Braden Highfield.
My name is Evie Monroe.
My name is Colin Tora. My name is James.
And Rayford Purdue, which is not here. He's part of this too. We are the 5 Direction team from Washburn School. The topic area we decided to work on was healthcare. Initially, we had other ideas related to radiation and energy. We decided to focus on healthcare because it was what seemed most realistic and helpful. In the system we designed should detect cancer cells and change their color when it's found. The input could be lymphoma cancer cells and the outputs could be a purplish bluish color. The next step to the design system is to figure out if it should be in pill form or be inserted. Some of the other ideas we thought about were a plant that is edible, that has all of the vitamins and can help with health and wellness. Some of the questions we still have are, would it actually detect well and how would it detect the cancer? What we liked best about, or we were most surprised about, is that it would help out with healthcare.
All right, great work, team 5 Directions. Cancer diagnostics is such a huge field, and I think absolutely things like lymphomas are super, super important to detect and be able to detect early. And I think, again, having like a nice clear output, something like a color or something that could maybe be detected in the body as well with either like an endoscope or maybe some sort of output that you can tell from the blood would be fantastic.
Yeah. I love it. Is incredible that these projects come up like within 3 weeks, right? Like these students were just starting to think about this just a few weeks ago, and they've come so far. And what I was struck by with this project, beyond, of course, everything you said, it's an important topic. There's lots of lymphomas. Early detection is critical. The idea that this one also has a purple output, like this is the very idea of abstraction that is so useful in engineering, right? It's like the input output. Our output is a purple color, and that purple color is going to come with different inputs, and that's how we expand the kind of biodesign space that we can be engaged in. So I think it's awesome. Yeah, congratulations! What a great project idea.
Absolutely, great work, team. And I like to include some of your other options as well. These all would be fantastic projects. All right, now next is the pink rhinos. How fun.
We are the Pink Rhinos team from Washburn School.
Here we decided to work on this project.
Initially, we had other ideas related to the environment.
We decided to focus on food because with food you have a huge variety of options, and we thought that we could come up with some extraordinary ideas to help better this area.
We got here by working together and testing information to see what we could come up with.
The system we designed to detect the pH level drop in terminal count.
The input that could be when pH levels drop, and the output could be when the milk will turn brown and tell the observer when it's gone bad.
The next step to design this system is to figure out how to take DNA from an oyster and a turquoise golden beetle to detect when the pH levels drop and to turn the milk brown.
Some of the other ideas we thought about changing the bacteria and weeds.
Some of the questions we still have are, how do you take the DNA from one organism and put it into another organism? And will our project design work and be able to be used?
What we like best about this work was getting to brainstorm any idea and understand how synthetic biology works.
Now we'll go to the slide. Experiment. Our design. We have decided to make a design that can detect when pH levels in milk drop and turn brown when you know it's gone bad. Sometimes expiration dates are not always right, but with this design, we can determine when milk has gone bad without the hassle of smelling and or pouring it out.
Things needed to create this design. Oyster. This is needed to detect the acid or the pH in the milk. Oysters are sensitive to acid, so they could easily detect the pH levels. Turquoise golden beetle these beetles can change to any color, but most specifically brown. So this would be a great help to our design. Milk. We would need milk to test this design. I would say we would need good milk and observe it until it went bad so we could see the design fully work.
Pros and cons. Pros. No need to have an expiration date. No sticking nose into milk. No more wasting food because of bad milk. Knowing when it's going bad. Cons. You can't use the milk after it goes bad for other cooking needs.
And that's it.
Awesome work, Pink Rhinos. Great, great, great job. Another nice, clear, colorful output. Fantastic. There's not much, honestly, in the kitchen that's worse than opening up milk and having to smell when it's gone bad. So if I could just look in and see, that would be phenomenal. That's a really, really great idea.
Yeah, I love it. I think it's amazing. I have written down now to figure out about those oysters and beetles. Like, I don't know about those phenomena that these students figured out about, but I think those are very cool and interesting. Yeah, I agree. And maybe there would be a way to like put this on the surface or something of like the caps. I don't know. Anyway, I just think that there's a lot of directions that this could go, and yeah, would be very useful.
Yeah, absolutely. I know it's so exciting to see teams turn to nature sometimes, because there's so much of biology that nature does really, really well. And being able to utilize sort of these existing technologies that have been developed by beetles and oysters would be so great for this project.
Yeah.
All right, and up next from DBXL, we have the BioBuds.
Hello, everybody. We're the BioBuzz. We're from DBXL. I'm Riley. This is Caitlin. That's Micah, and that's Eric. We also have Chase, but he's stuck in class right now. So our topic area that we are going to focus on is the environment, but other potential areas we were going to focus on was field health and another environmental idea.
So our final system design is a genetically modified E. coli that detects nitrogen compounds such as nitrates, nitrites, and nitrous oxide and produces a purple pigment color. We will do this using the PIRE promoter, which will create enzyme synthesizers, which synthesize enzymes that will react with tryptophan, a protein, to create the color purple.
So the reason that we're doing this is because we have a fish tank at our school that we're currently growing rainbow trout in. And in the past, the fish have died because of different, a couple of different reasons. One of those was probably being the nitrate and nitrite levels, because these can be detrimental to the fish if they get too high or too low. This would also help with a little bit of convenience so that we don't have to test it every day. So the reason that we're using E. coli is because after a lot of research, we found that E. coli is pretty easy to manipulate, and it also is a little bit cheaper, and it can grow pretty quickly. And the genes that we're going to implement will work in this organism.
So PIRE works as an inducible promoter. It produces VIO A through E via a chemical process.
So a couple of different things that we still need to figure out is how E. coli might respond to different stimuli and environments, and some issues that we might encounter with that whenever we're trying to implement the changes that we're trying to make to it. But once we figure that out, I think that it'll be a pretty good experiment for us to do. Thank you.
All right, fantastic job, Team BioBuds. That's great. You'll really be a bud to the fish, especially if this works out and can keep the water healthier. And at BioBuilder, we love nice purple outputs. We even have a lab that has a purple cell producing a purple protein as a key part of it. So great job finding even the specific proteins that you wanted to use for this. Really, really phenomenal work there.
Yeah, I think it is great. And yeah, purple is the color of the day, right? So I would point anyone who's interested in either reading more or thinking more about their projects to the BioBuilder Colorful World experiment, because as Michael alluded to, we can teach you how to make an E. coli that will turn purple. And that is right in that chapter. And then those bacteria turn purple all the time, but then you can think about how you would control the expression of purple. And just this week in the BioBuilder Club, we taught a little bit about how you make a genetic switch to turn something on or off. So all the pieces fit together, which is really very satisfying. And I think this project idea is super cool. It is a really incredible feat that all this can happen in just a few weeks of thinking about it. Congratulations to these students.
Absolutely, such great work. Awesome. All right, and time for questions. And I think first, just a huge round of applause for all of the teams again. This has been such great work. It's always so exciting to see how these projects turn out, and it's so impressive how much you've been able to do in just a few weeks. That really is not a lot of time to develop a project like this, and you all really pulled through and came up with such clever and creative ideas and researched them in all these different directions. Bravo. And thank you as well to our teachers that contribute to this, especially in such a sort of busy time of the year for them. We really appreciate all the work you put into this, making sure the videos get recorded and uploaded and the project material goes out on time. So another round of applause for our teachers as well. Thank you so much.
Can I give a shout out to the BioBuilder team? Michael, Kelly, you guys have made this this whole program work. Without you, there would be so many loose ends and so many things slipping through the cracks. Your expertise and your dedication to the success of the students and the teachers is really phenomenal. So thank you both for making this really hum and to get us to the point where we are today, which is incredible how fast the students have learned this much.
Well, thank you for your guidance and vision, also guiding all of this, Natalie. Since we have a couple students and other folks on the line, does anyone have questions for any of the teams or things that they'd like to add? Well, another thank you to our students, our teachers, and everyone. I'll go ahead and stop the screen share. But yes, fantastic job, everyone. Thank you so much for joining us. This has been a really great showcase.